Good morning, my dear students. Today, I would like to explain about the central dogma of DNA. I think already we have completed about the DNA and RNA, and also the types of DNAs and the types of RNAs and their functions. And now, in detail, we'll discuss about the individual functions of DNA. Before going to the process of replication, I need to introduce new terms. Those are DNA replication, DNA transcription, DNA translation. DNA replication means the conversion of dot parent DNA molecule into two daughter DNA molecules. So that means two identical daughter DNA molecules will be synthesized from one parent DNA molecule, that is called as DNA replication. Next, transcription. Transcription means the conversion of DNA into RNA. Translation means conversion of RNA into protein. So that means the RNA is in the form of a nucleotide sequence which is converted into amino acid sequence. That means the conversion of nucleotide language into amino acid language is called as translation process. So here comes the definitions: replication, transcription, translation. And another rare term you can see here, this is called as reverse transcription. Generally, the reverse transcription is very rare occurring process, especially in the virus like HIV. You can see the HIV virus can produce DNA by using RNA as a template. Here, the single stranded RNA can be used as the template for synthesizing the double stranded DNA. Next, the replication is maybe semi-conservative or conservative or random dispersive. In general, the DNA replication is semi-conservative process. So that means we can see in semi-conservative process, the two daughter DNA molecule will share a single strand from parent DNA molecule. So that means if it is conservative process, the two DNA strands from the mother cell will be shared as it easily into the daughter cell. But whereas in semi-conservative process, the only one strand of DNA will be shared with one daughter cell and another DNA strand will be shared with second daughter cells. But whereas in pure conservative process, both the mother DNA strands will be shared to a single cell and other DNA molecule will have two new DNA strands. The bidirectional replication. So here the replication process is bidirectional. That means the replication will start in both the directions. And first of all, the replication starts from unwinding the double-stranded DNA at particular point called as origin. So here, this is called as origin. Here, this area is called as origin, followed by synthesis of each strand. So in this, we can see the blue lines represent some mother strands, and pink lines they will represent daughter strands. And always, you can see the replication will starts from the direction from 5 to 3 direction, and this is in this direction from 5 to 3. The parental double stranded DNA and two newly formed double stranded DNA form a Y-shaped structure called as replication fork, and this is called as replication fork, whereas which has two parental double stranded DNA molecules and these two are newly formed DNA molecules. The replication fork in prokaryotes, it starts from the origin and forms a shape like theta. Next, come to the steps involved in the DNA replication. The first and foremost step is called as initiation. initiation of the replication starts by unwinding the DNA molecule. The enzyme called as helicase, which unwinds the DNA, forming a replication fork. So here, multiple replication forks along the DNA molecule creates replication bubble. Generally, it won't take. Rarely, it can take place. But in general, only one replication fork will be there in the DNA. The enzyme called as helicase. Next, second step is elongation process. In the elongation process, the new nucleotide molecules will be added in the direction of 5 to 3. Just remember that always the nucleotides will be added only in the direction of 5 to 3, but not in the direction of 3 to 5. Right. So addition of new nucleotides will be added in the 5 to 3 direction, and here the enzyme required for this function is RNA primase. It adds a complementary RNA primer to each template strand as starting point of replication, and this is RNA primer, which add a new template strand to the replication. So here we can see RNA primase is an enzyme, and also DNA polymerase is an enzyme. It reads the template strand from 3 to 5 direction and adds new complementary nucleotides from 5 to 3. So DNA synthesized in the direction of replication fork, it is called as leading strand. So this is considered as a leading strand. Means uh, the addition of nucleotides molecules, they are adding towards the replication fork. Here comes, you can see the adding of nucleotides is towards replication fork. So this is leading strand, whereas this upper strand is lagging strand. So in which the nucleotides will be added away from the replication fork. So that means from here to here, in this direction it will be added. That's why it is called as lagging strand. Next, and this is a lagging strand in which the nucleotide bases are being attached from away from the replication fork, and each strand that has been added in this direction, they are called as Okazaki fragments. And the DNA polymerase can only add new nucleotides in 5 to 3 direction, whereas we can see here also it is adding 5 to 3 direction, and here also it is adding 5 to 3 direction. And because of anti-parallel nature of DNA, a replication occurs in two directions. Because these two strands, we can see these two strands are in anti-parallel nature. One strand runs from 5 to 3 direction, and another strand runs from 3 to 5 direction. This strand runs from 3 to 5 direction, and this strand runs from 5 to 3 direction. Whereas this strand is leading strand, this strand is lagging strand. So, and each fragment is called as Okazaki fragment. And here comes with the enzymes RNA primer. RNA primase is an enzyme which can add a primer. Primer is nothing but the starting nucleotide sequence. And thereafter, DNA polymerase can add the nucleotide bases. Next, an RNA primer is laid down on other strand, and other nucleotides are added in 5 to 3 direction, moving away from the replication force, and this strand is called as lagging strand, and the segment produced is called as Okazaki fragments. Right, and later many DNA fragments are synthesized sequentially on the DNA template strand having 5 end, and these DNA fragments are called as Okazaki fragments, and these Okazaki fragments later they are joined, right, like this, and the DNA unwinds more, and leading strand is extended by DNA polymerase. So by keep on adding the nucleotides, this leading strand is synthesized continuously without any breaks. Whereas comes to the lagging strand, you can see the DNA polymerase is keep on adding the nucleotide, but the process is not continuous. And you can see in the leading strand, there is addition of nucleotides continuously, whereas in the lagging strand, the addition of nucleotides is discontinued. So we can see there is continuous addition of nucleotides in leading and fragments addition in the lagging strand. Next, termination process. And another variant of DNA polymerase which break down the Okazaki fragment and DNA, sorry, so a different type of DNA polymerase removes the RNA primer and replaces with DNA. And another enzyme, and this is very very important enzyme that is called as DNA ligase. It joins the Okazaki fragments and it forms a continuous chain. So now each new DNA molecule is rewound by helicase. Like initially we have seen the helicase enzyme which unwinds the DNA fragment, but whereas here again the helicase which will winds up the DNA molecules. And here comes to the summary of the DNA replication and facts about the DNA replications are remember the leading strand has one primer and continuous adding of nucleotides in the direction of 5 to 3 direction, whereas lagging strand it has multiple primers. That means for each DNA polymerase enzyme each primer will be added. That means for each nucleotide sequence one primer will be added, so that's why multiple primer will be there, and the addition of nucleotides is discontinuous. In humans the DNA polymerase can add up to 50 nucleotides per second. See how much speed, how much rapidly the DNA polymerase can add. So DNA polymerase can proofread its own work and also it does the excision repairs. That means if any error occurs in the addition of the nucleotide sequences, the DNA polymerase can proofread itself and also it can repair it. After proofreading, also unfortunately there may be uh, mutations may appear in the DNA sequences. Next, here comes we can see and the animation of DNA, and this is the replication fork, and this is the leading strand, and this is the lagging strand. So in the leading strand, initially a DNA RNA primer is added. So after addition of RNA primer, the DNA polymerase keep on adding the nucleotide sequences in this direction 5 to 3, but whereas and this is lagging strand, in lagging strand for each nucleotide sequence each RNA primer will be there. So it has multiple RNA primers and the addition of DNA nucleotides also not continuous, whereas the DNA ligase it will joins the both the multiple Okazaki fragments together to give rise a continuous nucleotide sequence. So the next step is transcription. Already I defined about transcription. Transcription is the process of conversion of DNA into mRNA. The genome of any organism contains all information for making that organism. The information is encoded in various types of genes that are transcribed into four types of RNA. So that is mRNA, it encodes the amino acids, it carries the codes. tRNA, it transfers the amino acids. rRNA, ribosomal RNA, it stores in the ribosomes. snRNA, small nuclear RNA, which is required for splicing. That means processing of unmatured messenger RNA. Initially, the messenger RNA is not ready to participate in the transcription, but whereas the snRNA will splice it and it will process it into pure matured messenger RNA. Right. The messenger RNA carries the genetical code that is useful in the synthesis of specific protein sequence. Next, the same steps will be involved in the transcription as we seen in the replication means initiation, elongation, termination. And first step is initiation. So generally, the initiation of the replication will take place at the replication fork, but whereas in transcription, uh, the initiation of the transcription will start from the area called as promoting area. That is called as promoter area, and this promoter area is identified by the presence of transcription factors, and these transcription factors are located next. The RNA polymerase binds to the promoter region, which is a region of bases that signals the beginning of a gene. RNA polymerase is bound to the Tata box of the promotion. This is called as the Tata box. Next step, elongation. And you should remember that transcription is the process in which a single stranded RNA will be synthesized from DNA, but whereas in the replication, double stranded DNA will be synthesized from DNA. Here in this case, only single stranded RNA will be synthesized from DNA, and that's why here only one strand of DNA will be used as the template. In this case, this strand is used as the template for the mRNA synthesis, and this strand, upper strand, is not used as this one. In general, the leading strand of the double stranded DNA will be used as a template for the mRNA. So here, there in the replication, we have seen DNA polymerase will move along the length of the DNA strand, but whereas here RNA polymerase moves along the protein encoding gene in the 5 to 3 direction, and it can add at least to 60 nucleotide per second, whereas DNA polymerase can add only 50 nucleotide per seconds. Next process is termination. You can see after addition of the nucleotides, the RNA polymerase enzyme reaches to the termination region, and at this termination region, the RNA will be removed from the DNA, and which is premature RNA. And here comes from the DNA through the transcription, a pre-mRNA will comes, and this pre-mRNA is not ready for protein synthesis. Just remember, this is not ready for protein synthesis. After processing of pre-mRNA into mRNA only, we can proceed for protein synthesis. And this processing is called as splicing, in which the regions called as introns will be removed and the exons will be added. The process of addition of these exons, both ends of exons, is called as splicing. And the splicing is done by small nuclear RNA, that means snRNA. Next, modification of pre-mRNA. At the fifth end, a cap is added to protect from exonucleases. So already we have seen about this. Next, splicing process. In the splicing process, the introns will be removed and the both ends of exons will be joined by using small nuclear RNAs, and they will form like peptides, and these peptides will splice out the intron and add the exons to give rise mature mRNA. Next, and right, so as of today, we have seen about the two procedures. That means replication as well as transcription. Just remember that replication is making of two daughter copies of DNA from parent DNA and making of mRNA from DNA, that is transcription. Replication and transcription are two different procedures that are involved. DNA replication gives rise two daughter copies. DNA transcription gives rise to one mRNA copy. Generally, DNA replication is useful in carrying the genetical information to the daughter cells, but whereas the transcription is useful in protein synthesis. We know about proteins. Already we have discussed in the macromolecules. Enzymes are proteins, hormones are proteins, some constructive proteins will be there, and some functional proteins will be there. Carrier protein molecules will be there. That means for synthesis of every protein molecules, there should be mRNA. But the mRNA has to come from DNA. For every protein, there is a specific gene is stored in the DNA that is called a genetic sequence or coding sequence. And from this coding sequence only, the mRNA will be synthesized as we seen in this class. That means in the three steps: initiation, elongation, and termination. Whereas in the DNA replication, DNA polymerase is the enzyme which would adds up the DNA nucleotides. Whereas in the transcription, RNA polymerase, it will add up the nucleotide sequences. And another fact is always the nucleotides will be added in the 5 to 3 direction. Always any polymerase, it may be DNA polymerase or it may be RNA polymerase. Always it will add on into 3 direction. In the DNA replication, the two strands will be used. Both strands, leading strand as well as lagging strands will be used. Whereas in the leading strand, the addition of nucleotides is in continuous direction, whereas in the lagging strand, it is in discontinuous addition. That fragments are called as Okazaki fragments. Later, they will be joined by DNA ligase. But whereas in the this transcription process, there only one strand will be used, that is a leading strand, and also the RNA polymerase will add up the nucleotides. And I think you know about there is a difference in the nucleotides between the DNA and RNA. Right? RNA has uracil instead of thymine, so that's why here the RNA polymerase will add the nucleotides. Right. So immediately the pre-mRNA will not use it for synthesis of proteins. So then it has to modify it into mRNA through the process of splicing. So which is done by small nuclear RNAs. So dear students, now I would like to end my session with this, and in the coming class we will learn about translation. That means how this mRNA will be converted into, and that means the sequence of mRNA which has nucleotide sequence is converted into amino acid sequence, that is in ribosomes. Ribosome, since it is a protein synthesizing machinery, and this mRNA, which is a product of transcription, enters into the ribosomes, and according to the sequence that is present in the mRNA, that means some nucleotide sequence may be there in this mRNA. Depending upon the sequence that is present in the mRNA, proteins synthesis will take place in the ribosomes. So that means translation. We'll discuss about the translation in the upcoming class. Thank you. If you have any doubts, you can contact the WhatsApp group. Thank you all.